Thank you, dear Chairman. And thanks to the previous speaker for raising these interesting questions. Thanks for the Academy for inviting me. I would really love to discuss some of the questions raised, but my presentation will focus on emergency contraception and mechanism of action. I think part of the answers to the questions raised is that we need to collaborate with pharmaceutical industry. If there is no company, it's very difficult to introduce a drug. I try to collaborate with as many companies as possible because I think contraceptive development is extremely important. Now, the definition used for emergency contraception is any method used after an unprotected intercourse to prevent an unwanted pregnancy. And clearly, we need such a method which is clear in the case of sexual violence, for example, but also just common human mistakes to help women to avoid an unintended, unwanted pregnancy and also maybe an abortion. And I love medical history. This is the first whole chapter actually on contraception in the first textbook on contraception or gynecology written by Soranus of Ephesus. And I think we can see here that he described a post-coital emergency contraceptive method. So how do we get rid of the sperms? Today, I've listed the method used today. We still have the USP method in countries where there are no other alternatives. And today, since we don't have high-dose pills, it may mean to use 8+8 or even 10+10 combined pills. LNG, the gold standard sort of is 1.5 milligram single dose. Then we have UPA, LR1, 30 milligram single dose in an increasing number of countries. And mifepristone, as we just heard, in some countries. We also have the kappa IUD, which is actually the most effective method. But I was asked to talk about hormonal methods. We heard about the WHO trials, and there were 2 large WHO trials from the expert group where we were both members. And the first one was comparing use with LNG, and the second one also included mifepristone, as we heard. And these trials really changed the landscape of emergency contraception. Now there are several possible targets, of course, for emergency contraception if we just look at reproduction. And I think it's crucial to have the knowledge to understand the window of fertility when we discuss this. I think that's frequently a mistake that people know that they don't understand these basics. So we have a fertile window where an intercourse may result in pregnancy. And here you can see the fertile window above the picture of the menstrual cycle. And just to remind you, of course you know, but it's frequently misunderstood that actually an intercourse during the luteal phase will not result in pregnancy. And then, of course, it's another thing that have a method that would block endometrial function would be effective method to block even a fertilized egg or an embryo from implanting. But we need to keep these differences in mind. Now, if we look at the effects on the sperms can be retrieved from the endocervix within 90 seconds and from the fallopian tube within 5 minutes of an intercourse or insemination. So I think it's clear that this would not be a main mechanism of action of an emergency contraceptive method. Then it has to be really emergency. We also have data from looking at LNG in doses relevant for emergency contraception and also mifepristone. And actually no effect on sperm function in vitro. And the same for ulipristal acetate in the dose used for emergency contraception. So no effect in vitro. There is only one study looking at UPA on sperm DNA fragmentation in vitro, where there is an effect. And whether that has any clinical significance, we don't know. But what was shown here is that UPA had an effect on oxidative stress and prevention of DNA fragmentation of the sperm. It's well known also from the classical studies in the 70s by Kisuru that levonorgestrel, as are the gestadiens, will have an effect on cervical mucus. But again, this effect comes later. So when LNG was given 3 to 10 hours post-coital, there was a reduced number of sperms in the uterine cavity, immobilization of sperms, increased viscosity of cervical mucus, and changed composition of uterine fluid. But these changes occurred later, so they're crucial for regular use of gestadiens, but not for emergency contraception. Now, we and others have also looked at the effects on follicular development and ovulation. And the model, sort of in vivo model used is frequently to recruit fertile women with proven fertility to administer levonorgestrel 1.5 milligram pre or post-ovulatory, to include control and treatment cycles and washout cycles in between, to use daily ultrasound examinations to monitor follicular development and ovulation, to obtain endometrial biopsies at the expected time of endometrial receptivity to look at endometrial changes. And also, we've also looked at fallopian tube actually in a similar way in women undergoing tubal ligation. And then to monitor daily steroid hormones. And when we and others look at the effect on LNG in doses used for EC on follicular development and ovulation, again, the effect depends on when LNG is administered. So if it's used after selection of the dominant follicle, but before the onset of the LH peak, or the LH starts to rise, we can see variable effects on follicular growth. It can mean a delayed development, inhibited growth, or an unruptured follicle. And if administered after the LH onset, it's actually not effective to block ovulation. And here is just one figure showing controlled pre-ovulatory administration and post-ovulatory administration and the effect on the LH surge. Taken together, sort of doing a meta-analysis of studies looking at this, we can see that LNG, as I said, administered at a follicular diameter of 12 to 70 millimeter, will result in follicular rupture within 5 days in only 44% of cycles, and in 30% of cycles, rupture will be delayed for more than 5 days. In 26% of cycles, the dominant follicle will become an unruptured luteinized follicle. So this adds figures to what I said before. Now, if we look at the effect of UPA during the menstrual cycle, 30 milligrams as used in ELA1 or ELA. In the mid-follicular phase, it will inhibit or rather delay follicular genesis and steroidogenesis. And if it's given at LH onset or after LH has started to rise until the LH peak. So the window of action of UPA compared to LNG is wider because there is an action up to the LH peak, while for LNG it's up to the onset of LH increase. But the effect will be the same to inhibit follicular rupture, and this is through effect on progesterone receptor A. Now, there have also been attempts to improve the efficacy of LNG. And one way that we also discussed frequently in our WHO research group was to add a prostaglandin endoperoxide 2 inhibitor. They were previously called COX-2 inhibitors. And meloxicam is such a compound. The hypothesis is that it will act in synergy with LNG to prevent ovulation. And we know that prostaglandins are involved in follicular rupture and ovulation. We know that knockout mice knocked out for COX-2 inhibitor will be infertile because of anovulation, but they have also an effect on endometrium to prevent implantation. So this may result in a more effective method. Now, if we look at pooled analysis of 3 studies, and they have all been conducted by Horacio Croxatto from Chile and also Vivian Brache from Dominican Republic and others working with Population Council. The first study is LNG versus placebo. The second one, LNG versus LNG plus meloxicam. And the third is UPA versus placebo. And here it was shown that persistence of dominant follicle post-treatment when LND was compared to placebo, there was no difference. LND plus meloxicam was more effective than LND alone or placebo. And persistence of dominant follicle according to the LH at treatment, if administered before the LH peak, LND and meloxicam resulted in 45% of cycles or dominant follicles present 5 days after treatment, so delayed ovulation. And LND versus placebo, again, no difference. And for all, administration after LH peak, no effect. Then it's too late to block ovulation. And here you can see the results in the figure. Here, again, and of course that may also depend on the doses used, but UPA seems still to be the most effective here to prevent follicular rupture within 5 days after treatment. Here is a survival analysis of time to follicular rupture from treatment to the 5th day after treatment, and again by treatment group. And you can see the same thing again, UPA more effective and very little difference between LNG and placebo. Now, if we look at LNG administered post-ovulation on LH plus 2, in animal studies, rat embryo transport is regulated by estradiol and progesterone. LNG after ovulation did not prevent pregnancy, and the same was observed in monkey studies. LNG after ovulation had no effect on pregnancy. And we have looked at the effect on fallopian tube. We know also in humans that progesterone regulates tubal transport in vivo and in vitro, muscular contractions and cilia activity. And exposed to high doses of progesterone will result in a lower frequency of cilia activity, which can be reversed by mifepristone. So what we found was that when exposed to doses of LNG or mifepristone or UPA in doses relevant for emergency contraception, there was no effect. And this is also consistent with data from 136 studies on mifepristone or LNG emergency contraception, showing no increased risk for ectopic pregnancy compared to the rate in the general population. Now, LNG post-ovulatory and the effects on the endometrium. So, and remember, this is a single dose, 1.5 milligrams. There was no effect on endometrial histology, no significant effect on markers of endometrial receptivity, and actually the same results with a repeat dose of LNG or when administered vaginally. There are also, of course, what's popular now to do microarray studies and looking at changes in gene expression during the receptive period. And neither the magnitude nor the nature of these changes observed after LNG exposure would indicate any interference with endometrial receptivity. With UPA and endometrium. There are a few studies looking at and showing a dose-dependent effect, which would not be surprising based on all the knowledge we have with mifepristone. Histological evaluation, it has looked at endometrial dating and thickness, which we know is very actually poorly correlated with receptivity and fertility. And there were only a limited number of receptivity markers studied up to last year. We have, of course, it's very difficult to study human embryo implantation, and we can't do it in vivo for natural reasons. So we have developed an in vitro model to be able to study human embryo attachment at least. And we use endometrial biopsies, timed biopsies, where we separate the cells. We build a 3-dimension endometrial construct, and you can see here that it looks very similar to the natural endometrium in a cross-section, with stroma and glandular formations and luminal epithelium, and also with polar, the cells maintain their polarity, which you can see here, epithelium and stroma. And here we can study embryo implantation with surplus embryos from our IVF program donated by couples. We know from mifepristone that it's, as I said, the effects are dose dependent and dependent on when mifepristone is administered. And following treatment in the follicular phase, a low dose will result in delayed follicular development, which will then resume, resulting in a normal ovulation and normal endometrial development. A higher dose will block follicular development and ovulation, and it's not until a new follicle is recruited that the cycle will resume. And post-ovulatory treatment with a high dose will, as I showed yesterday, will affect endometrial receptivity and will be highly effective to prevent implantation. Now, when we looked at a high dose of mifepristone, 200 milligrams on LH+2, which we know is effective for contraception, and compared to controls and levonorgestrel in our in vitro model, and it's important to remember, of course, not 100% of Blastocyst will implant. That would not be physiological. But we can see here, when we compare embryos implanting in the control to levonorgestrel, there is no significant difference, while a high dose of mifepristone will completely block implantation. We also looked at lower doses of mifepristone in the implantation model, and it's clear that there is a dose-dependent effect. So the lowest doses would not have any significant effect, while a higher dose will block implantation. So a clear dose-dependent effect depending on the effect on the endometrium. We have also, as others, and what is of course the trend now, looked at microarrays to look at the changes in gene expression. And there are some changes, but actually not well correlated to, there can be smaller changes without affecting receptivity and implantation. Now, what happens with UPA in a dose relevant for emergency contraception? There is a recent study actually published this year when they administered UPA 30 milligram at follicular diameter 20 millimeters, and then took an endometrial biopsy, and they included 12 women. They did microarrays and PCR analysis, histology and immunohistochemistry, and they found that they had 47 genes changed, up or down regulated, compared to control. This is consistent with our finding. When we look at UPA, we can also see that there is an effect on some of the genes, which is, again, no surprise. We looked at known receptivity genes. There were actually only 2 genes that had a significant upregulation, and 4 were significantly downregulated. But most important, when we looked at the effect on attachment and implantation, actually it was not significantly affected. So despite these changes, and we know that the microarrays are extremely sensitive, it depends on the day you take the biopsy and so on, but they have very little clinical significance. When we look at the effect of mifepristone or levonorgestrel or UPA on the embryo development in this in vitro model, there is no direct effect on the human embryos. So they are still viable and can implant in an endometrium which has not been exposed to any of the treatments. We know with LNG and also the little data that's available for UPA or low-dose mifepristone that there is no adverse outcome of the pregnancy. We also have supportive clinical data. There are a few studies looking at administration of LNG before or after ovulation, and showing that women using LNG before ovulation has an effect of the treatment, while post-ovulatory treatment could not inhibit pregnancy. And actually very similar results from a recent study published last year on UPA exposure, again effective when administered before but not after ovulation, which supports, of course, that there is no post-ovulatory effect. Also, the clinical data looking at unprotected sexual intercourse, women who participated in the clinical trials on emergency contraception, if they had their intercourse after LH peak at the time of ovulation, they were not protected, so the treatment was more likely to fail. And also women who had used emergency contraception but had subsequent unprotected intercourse later on during the same cycle, because delayed ovulation but no effect on the endometrium, and then no protection. What I proposed yesterday, endometrial contraception, would of course be a means to improve the efficacy of emergency contraception if we had an additional effect on the endometrium. But that's for the future. Now, it's clear that the oral pills and the doses used today, LNG and UPA, and also low-dose mifepristone, are not at all as effective as the copper IUD, because the mechanism of action is more limited. The window of action is much more limited. I also added a few slides on clinical recommendations, because it also has to do with the mechanism of action. And one problem, of course, is interactions, since these are steroid hormones interacting with the progesterone receptor. So for women who use enzyme induction drugs, which could affect the efficacy of the pills, kappa IUD should always be the first choice. There is a recommendation from the EMA to double the dose of LNE in these cases, although we have no clinical data actually supporting that would be more effective. The efficacy of UPA-EC of LR1 can be reduced by drugs which increase gastric pH. I think that's important to remember. And even more important, by a gestagen. So previous use of a gestagen within 1 week prior to UPA can reduce efficacy because they bind to the same receptor. And also, if UPA is used, it's recommended to wait 5 days before using an hormonal method containing a gestagen, because again, otherwise an interaction at the level of the receptor. So can the use of emergency contraceptive pills be repeated in the same menstrual cycle? Yes, They can and should be repeated as needed. Of course, we have more effective methods, which would be smarter choice, but if needed. There is an increased risk of irregular bleeding. There is, and important to remember, so if needed and if there is more than 24 hours from the use, a new dose of EC should be recommended. And which ECP should be recommended? If LR1, ulipristal acetate, has been used, LNG should not be taken within the next 5 days because of the interaction. If LNG has been used early in the cycle, LNG can be repeatedly used. There is no interaction. And if a woman has already taken LNG, UPA should not be taken in the following 7 days. So this is a little bit tricky but important to remember. So what about quick start? We recommend that women should quick start a more effective method immediately after the use of emergency contraception. If she has chosen a cup IUD, of course she has already protection. But otherwise, since the mechanism of action includes postponing ovulation, if she has further acts of unprotected intercourse, she may be at risk of a pregnancy. So with UPA, she should wait 5 days before starting an hormonal method and use barrier method until her method is effective, that is 1 week. And for LNG, quick start is possible, but again, barrier method until the method, started method is effective. Because so as I try to explain, the hormonal method may interact with the efficacy of UPA, but UPA actually has no interaction with the efficacy of the combined pill, which is also important to remember. So in conclusion, the mechanism of action of emergency contraceptive pills, LNG or UPA, is mainly due to impaired ovarian function, follicular development and ovarian ovulation. UPA EC is effective after LH has started to rise until the LH peak, while LNG is only effective up to the start of LH increase. And they have no significant effects on the endometrium in the doses used for emergency contraception. And none of them, in doses used for emergency contraception, inhibit implantation in vitro and has no adverse effect on the embryo or on pregnancy. So thank you.